Welcome, ladies and gentlemen. So today we're going to talk about Chapter 24, which really is just going to get into the mechanisms by which we get an existing species to sort of branch out or create new species as we move forward in evolutionary history. So what we're really going to talk about here is this concept of speciation. And speciation just is defined as the origin of a new species. Okay, well, we need to back up a step here, though. If we're going to talk about the creation of a new species, we need to have some commonality in defining what a species is. So, species actually comes from the Latin word meaning kind or appearance, and biologists use all sorts of data to define what a species is. But if we were to just take a textbook definition, it's generally defined as a group whose members possess similar anatomical characteristics and have the ability to interbreed. And so we take into account morphology. What do they look like? What are they structurally defined as? Their physiology, how do they function? Their biochemistry, their DNA, all of this data collectively we use to compare one set of organisms to another and define whether or not those two groups are the same species or something different. So ultimately, speciation is the crux of evolutionary theory. It's the way that we discuss changes in organisms over time and the overall biological diversity. This is the source for it. So evolutionary theory has to be able to explain how new species originate. How do they come to be? And so we can look at it from a macroevolutionary scale, changing at the big species level, which is what we're talking about here. And if you go back to Darwin, that's really what he was focused on. These were these big overarching changes in the species level that he was able to study, stuff we can see at a macroscopic scale, as opposed to looking at microevolution, which is what we were talking about in chapter 23, where it's just the genetic makeup of the population that is changing over time, as opposed to these big fundamental changes that we can physically see. Macroevolutions on a microscopic scale, we're talking about the genetic level instead. So when we look at speciation, we really kind of dive into these two major patterns. Either we can talk about evolution occurring in this process of anagenesis, which is just the accumulation of heritable changes that ultimately alter the ancestral species into the characteristics of the species we see now. You may come across anagenesis as phyletic evolution. What you're probably more familiar with is the cladogenesis, or the branching evolution, where we get new species that arise from a single ancestral species. So we branch out. We create more diversity as a result by increasing the number of species from those branching points. So let's go back to the fact that if I'm going to create a new species, that means I need to have a definition by which that species is defined. So we look at groups of populations whose members in that population have the potential to interbreed in nature and produce viable offspring. This is the pretty common definition of what a biological species is. So interbreed, produce viable, fertile offspring. That is the overarching idea behind this biological species concept. So individuals who can't successfully breed with each other must be of other populations, and as a result must be a different species. So long as populations are capable of maintaining gene flow between those populations, meaning individuals can move in and move out, and they can continue to mix up their gene pools by successfully mating and reproducing with each other among those populations, then that keeps the phenotypes of those populations together, and as a result maintains those two subpopulations still being of the same species. So biological species concept really emphasizes this idea of reproductive isolation, meaning that something along the way allows those two populations to become so diverged probably at a genetic level, that they can no longer successfully mate and produce viable fertile offspring as a result. So where does that reproductive isolation come from? Well, it's usually some sort of geographic isolation that starts it, although that's not necessarily required, where those two populations physically become incapable of accessing each other. Not always required, but that's often what we see. That's a lot of the classic examples of things that we'll see that ultimately lead to new species. They can't get to each other, environments are different enough, and they stay separated long enough that even when that barrier is removed, they've reached this point of reproductive isolation and they can no longer get to each other. If that's not the case, there are other instances where that can happen, whether it's behavioral, timing, all sorts of things that we're going to take a look at. But point being, we get to the point of reproductive isolation, however we've gotten there, and that reproductive isolation preventing the production of those fertile offspring now allows us to start talking about two different species. That being said, it has to not only be viable offspring, but fertile offspring. So there are instances where we can still get two members of different species mating together, creating a hybrid. But if that hybrid isn't fertile in and of itself, then that perpetuation down the line doesn't happen. So reproductive isolation really gets classified by a handful of factors that either occur before or after fertilization. And that's where these terms pre-zygotic and post-zygotic come from. So remember, a zygote is a fertilized egg. So if we're talking pre-zygote, those are your pre-zygotic barriers, so they're going to happen prior to the fertilization of egg and sperm. Post-zygotic would be the ability for that egg and sperm to find each other, and then some sort of barrier prevents the success of a viable fertile offspring post that fertilization process. So prezygotic, this actually blocks the fertilization from occurring at all. There's a handful of ways this can occur. It can be impeding different species from even attempting the act of mating. It may prevent the successful completion of that mating, or just may hinder the fertilization even if the mating act is capable of happening successfully. So a lot of times we're talking about things like this. So habitat isolation, for example. This is a pretty common one for kids to wrap their heads around because we literally have them occupying two different habitats. And so they just don't really ever connect with each other. This is that idea of starting off as geographic isolation and then staying physically barriered or barred from each other long enough that the reproductive isolation comes with time. Temporal isolation is a matter of timing. This one happens a lot when we look at plants. So species that breed at different times of day or different seasons or different times of year just naturally will never get their gametes to mix because they're just not present at the right times. So looking at flowers blooming at different points in the year would be an example of temporal isolation. I can't have two flowers mating if they bloom at different times. The whole purpose of that flower is the pollen, is the semen, the egg is within the flower structure itself. And if they can never get to each other because they just don't produce those gametes at the same time of year, then we're not going to be able to have a successful offspring as a result. Behavioral isolation, this happens a lot with sexual selection, and we have specific courtship rituals that attract their mate, and those behaviors are often unique to individual species. So if a female bird is expecting a certain type of mating dance and doesn't get that from the male of another species, then she's more than likely not going to bother mating with him. Mechanical isolation, these are physical morphological differences. They physically can't complete the act of mating because the parts don't fit. gametic isolation, the sperm of one species just may not be able to fertilize the eggs of another. So although that in that situation, the two individuals may actually be able to complete that mating act, once that sperm is released, the either biochemical environment of the female or just the biochemical molecules in the egg to sperm just aren't compatible, and so they're not able to actually allow that sperm to penetrate the egg's membrane, and so the fertilization never happens. So post-zygotic then works on the opposite end of it. So they're able to successfully mate, but now we're looking at some issue along the way with the offspring itself. And so we get these three potential mechanisms that occur. So reduced hybrid viability. The genes of the parent species can interact, but they impair the hybrid's development somewhere along the way. So we may get an embryo to form, but that embryo may never successfully reach delivery, for example. Reduced hybrid fertility. Hybrid can be incredibly vigorous but sterile. This is your mule example. So you get a horse and a donkey to mate, they produce a mule. The mule is an excellent work animal. By all other indicators, should have incredibly great fitness, but because the definition of evolutionary fitness is reproductive success, the mule is completely wiped out of that race. Its fitness is zero. It can't reproduce. So although it can survive into adulthood and be a very successful, long-living offspring, when it goes through the process of meiosis to make gametes, that process fails. And it really comes down to the number of organisms, the number of chromosomes in that organism that aren't able to split out appropriately, and as a result, that structure doesn't end up producing viable eggs or viable sperm. Hybrid breakdown is an interesting one because this is going to take multi-generations before it fails. This is probably still not a complete reproductive isolation, but we're getting darn close. So you get that first generation of hybrids. So two different species, male and female, will mate, they'll produce the offspring, that's your hybrid. That hybrid is viable and capable of producing successful gametes. We mate those two first generation hybrids together, they can successfully mate, but it's those second generation offspring then that are either just weak in general in terms of their viability, or they're sterile, or both. And so we never make it past that second generation of hybrid to actually discuss having that hybrid being its own species. And as a result, backtracking to recognizing that those two parents are in fact separate species. So this is a nice figure in your textbook. Take some time to look at it, but it'll talk you through and give you some examples of the different types of pre- and post-zygotic barriers with some actual examples. So here's the kicker. We're using this biological species concept and talking about these pre- and post-zygotic barriers, which is useful, but there's some breakdown in the way that this concept works. So when we talk about using the biological species concept to define what a species is, I'm not going to be able to apply it in every situation. I can't apply the biological species concept to organisms that don't sexually reproduce. If I'm an asexually reproducing organism, then how do I tell whether or not I can successfully mate and produce viable offspring? I am my mate. And so this concept isn't going to be useful for them. It's also not incredibly useful when we talk about applying it to fossils because they're fossils and they're presumably extinct organisms. And so I'm not able to watch their ability to mate in nature and find out what happens. And just organisms in general that we don't really have a well-known understanding of how their reproduction occurs, and there are definitely organisms that is the case. So we can't easily apply the biological species concept in those situations. So overarchingly, the biological species concept emphasizes the absence of gene flow. So we prevent these organisms from successfully mixing up their genes and passing it on. However, there are situations where gene flow can occur, in rare situations, mind you, between very distinct species. So we know that a grizzly bear and a polar bear are defined as separate species, and yet their genetic makeup is close enough that they can successfully mate. It just doesn't happen often. And so, how do we reconcile that with this idea of the biological species concept? And so, there are other definitions for what a species is, and oftentimes, when biologists are defining what a species constitutes, if this species is something different from that set of organisms, they'll utilize multiple pieces of data above and beyond just their reproductive habits. So that's where some of these other definitions of a species may come into play. We can talk about morphological species concept, where we're utilizing characteristics to determine a species based on its body size, its shape, other structural features. This one can definitely be applied to both sexual and asexual species. But just like anything else, it also has its own drawbacks and relies on a lot of subjective criteria. So go all the way back to Carlos Linnaeus when he's building this first initial classification scheme, or even Darwin for that matter, back in the 1800s, looking at these organisms, all you have to go off of is physical characteristics and your ability to differentiate one from the other. And what I may say is definitely two distinct features. Somebody else may say, nah, they're close enough, it's probably the same thing. So it's much more subjective. Paleontological species concept is much more useful for looking at fossil records, but again, focuses mostly on those morphological, discrete features. Again, gonna be able to apply to both sexual and asexual species, but it's difficult to determine the degree of difference for separate species in the fossil record because you only have some subset of that population that gets fossilized in the first place, and then you've got to find it. And then once again, you're still subject to objective criteria. Ecological species concept is another option. Here we're viewing the species in terms of its ecological niche. So again, useful in both sexual and asexual species, but it emphasizes the role of disruptive selection. So what those organisms are capable of doing in their environment and how that can branch off from what they're truly capable of doing versus what they actually do. So sort of that idea of potential versus realized niches can get a little messy there. Phylogenetic species concept. This has become much more favorable the more that we are capable of doing genetic research and sequencing genomes and understanding all of that. The more that that's getting easier, more accurate, more reliable, cheaper, faster, all of that. This has kind of been the route that we've been taking much more recently. And phylogenetic species concepts really are looking at the organisms within their own unique genetic history. And so we have to have the ability to understand what genes are, what they do, how to sequence them. And again, we're getting better and better at that and comparing that